Começou.
Okay. Well, good morning, good afternoon. Yeah, it's morning still. To everybody, thank you for being here today at this fourth webinar of the AquaInvert project. Today, the theme of the webinar will be centered on grow out issues for the different invertebrate species that are being studied within the AquaInvert. And we're going to have some speakers from the University of Las Palmas in Gran Canaria, Spain, from the University of Azores, and from Arditi in Madeira. So welcome everyone, and I'll introduce now the first speaker. Her name is María del Pino Viera Toledo. She's from the Aquaculture Research Group from the Aqua Institute in the University of Las Palmas. And she's gonna present different works that have been done on Haliotistubercula cocinea on the island of Gran Canaria, and centered on the grow out issues. So Mappy, the floor is yours.
Thank you. Empezar de nuevo. Sorry, I did something wrong. So now, no? Sorry for the delay. Good morning, everybody. Thanks for giving us the opportunity to present part of the work program that we have undertaken during almost last 20 years to develop the culture technology, in grow techniques of our local abalone species, Haliotistubercula cocinea. But just to introduce this mollusk, the abalone are marine one-shell gastropods that belong to family Haliotidae. They are 56 currently described species. They all belong to just one genus, Haliotis. And they have a worldwide distribution. They are benthic animals, most of them located between 3 and 10 meters deep, where the macroalgae, which is their natural food, is more abundant. They present a separate sex. Mature males and females can be easily recognized by the difference in the color of the gonad, as you may see in that picture. And tropical and subtropical species are smaller, grow faster than those in temperate regions that can reach up to 30 centimeters in shell length. Abalone have traditionally been highly prized delicacies worldwide. Its flesh is used for food or also even cosmetics. And its shell, which is quite nice, it's used for implements or decoration. As with other seafood, production has shifted from wild caught to farmed, and today over 95% of the abalone comes from aquaculture. This increase being possible first by the development of production practices, both for juvenile stages and for on-growing systems. Among all abalone species, 15 are globally exploited, with the Haliotistubercula, which name is Pacific abalone. It's the preferred one. And of course, China is the largest world producer. In the Canary Islands, in the Micronesian, there is the species is Haliotistubercula cocinea. It's a subspecies of the European wormer, but it is a little bit smaller. It just grows to a maximum size of 8-9 centimeters. And in the Canary Islands, it has been locally exploited for decades, but leading to the an over-exploitation of its stock, which are almost depleted. Hence, there is public interest in developing the culture technique of these species, not only to supply the local market, but to try to contribute to the recovery of the wild stocks. Our previous studies focusing on reproduction, early life stages, and those that I'm going to present now regarding on-growing, suggest this species as a good candidate for the Canary aquaculture diversification. A proper growth techniques and the resulting growth of cultured animal, not only growth, but also survival, depends on many factors. Culture condition, grow out system, sea base or land base, etc. But mainly those regarding feeding and nutrition, feeding patterns changing during the different stages of their life cycle. Feed option for growing abalone can be based on the use of wild seaweed, which nutritional composition and abundance varies greatly, or formulated feeds that can be, that they are formulated for each abalone species. No? Since white algae are not commercially valuable in the Canary Islands, the development of the abalone industry should be reliant on the use of culture macroalgae and or formulated feeds. The treatment of aquaculture wastewater and the mitigation of the potential environmental impact of aquaculture can be achieved through the implementation of multi-trophic aquaculture system that and the seaweeds production being an added income as feed for shellfish such as abalone. Let's say to be harvested. We have undertaken several studies just to try to develop our the culture technology specifically for our conditions in the Canary Islands. So in the first study, we conducted a 60-day feeding trial to assess the suitability of 3 red algae, Ipnea spinella, Gracilaria cornea, and Isnea musiforme, for growing juvenile abalone. The initial size was just 11 millimeters long. And the approximate composition of that macroalgae revealed that they all satisfy the abalone nutritional requirements. What we got was that the growth rate of abalone were in the range of those obtained under commercial condition. However, feeding Gracilaria cornea led to the lowest, sorry, I'm having with, okay. Led to the lowest growth performance, not related with its nutritional value, as we saw, but to the lower feed intake related to the harder texture of this macroalgae. Because you may see that both food conversion ratio or protein efficiency ratio of this algae was similar to Ignea spinella, that was the best one. So this study, the first one that we did with this macroalgae, revealed that they all have a good potential for successfully produce the abalone, but among them, Ignea spinella showed the highest value. Then we went further and we evaluated the growth performance of juvenile reared in an IMTA system compared with that of abalone fed non-enriched macroalgae. For macroalgae treatments, we were evaluated 3 monospecific, Ulva rigida, but in that time we introduced a green algae, Ignea spinella and Gracilaria cornea, and a composite one of those 3. And 4 were reared in the IMTA, and the other were reared in the fresh sea water. The study lasted 3 months. As you may see in the graph, seaweeds reared in the fish pond wastewater effluents increase on at least double their protein content. And of course, animals that ate this enriched macroalgae, they perform quite better than those fed non-enriched macroalgae. Similarly, animals that fed the mixed diets perform significantly better than those fed single algal diets, indicating that essential nutrients may become limited when they are fed with just one type of algae. So the results of this study clearly indicate that Haliotistubercula can be efficiently grow out in an integrated culture system, suggesting that on-farm seaweed abalone production could be a part of future or potential development of abalone industry in the Canary Islands. To date, considering the nutritional variability of white macroalgae and the need to limit the use of fish meal in compound feed for ecological and sustainable abalone aquaculture, we conducted a 16 weeks trial to evaluate the suitability of different seaweed meals, included alone or in combination with fish meal in formulated diets. In this one, we tested Palmaria, Porphyra, Helidium, and the one that has Palmaria also included 9% of fish meal, because this was like our control diet, because we had tested it in a previous study and against including laminaria, but this was quite better, so we decided to use this diet as a control. All of them has also ulva and gracilaria, or let's say hydropuntia. We can use both names. We tested 2 control diets, a commercial one, which is the European aloe from Scretin, and the enriched fresh ulva and gracilaria. This is the approximate analysis on the water stability, which is a quite important factor in abalone diets, because it has to stay stable, or it should be stable at least 24 hours. We feed them with diets daily, whereas when you feed with the macroalgae, you can feed them weekly or even monthly. Okay, so overall, the enriched fresh algae demonstrated a significantly higher dietary value for Haliotistuberculata than the compound feeds. However, this is not, it was not significantly different from the one that has Palmaria and 9% of fish meal. Anyway, feeding along with the fish meal-free diets, the other three, promoted high survival, sustained growth, and good protein utilization. The inclusion of Palmaria palmata improved growth, whereas the opposite was attained with where Porphyra or Helidium were included because we obtained a lower growth and protein efficiency ratio and a higher food conversion ratio. As you may see, the overall growth between 76 and 112 was a quite good growth for abalone, even higher than those obtained in commercial diets farms that can be, average 80 microns per day. So I have a stocking factor, stocking density and culture system, they are quite important factors to grow abalone. So we, in this occasion, we evaluated the effect of different stocking densities on the growth performance, feed utilization, and of course survival of two initial different sizes groups, and 30 and 40 millimeters abalones during the final grow out to cocktail market size, which is between 45 or 60 millimeters in shell length. So for this purpose, trials were performed in an abalone cages that we installed in a commercial open sea cages fish farm. And the name of these kind of these devices is Ortax. They come from an enterprise in New Jersey in United Kingdom. Overall, we obtained a sustained high linear growth during the whole trial, both for shell and weight. However, between 17 and 19 reduction in weight gain was obtained by doubling the stocking density, suggesting a higher competition for space or food, which has been recorded before in many studies with other abalone species. Nevertheless, the high growth performance that we obtained and the survival that was very high also, even at high densities, denoted the suitability of the offshore aquaculture system, as well as the biofilter produced macroalgae for grow at Haliotistubercula cocinea. That overall, we think that we saw that can reach the commercial size in just 18, 22 months. Thank you.
Thank you very much, Mappy. We've seen that, I mean, there are some question on the chat, and as we've done previously, we will keep the questions for at the end of the presentation to have a global conversation between the members of the audience. Okay, So next presenter now is Diego from the RDT Center in Madeira. Diego is focusing his research on the production of patella, and he's done many interesting experiments on the larval production and post-larval production of patella in Madeira. And today also he's gonna focus his presentation more on the grow out subject, although we know with patella it's a bit more tricky, and then the research is still pretty ongoing. So thank you very much, Diego. The floor is yours. And again, if anyone has questions, please put them on the chat and we will look at them and discuss them later on. Thank you.
And thank you very much, Jason, and welcome to everyone for this last session of this webinar series. I think that's everything well. Well, in this seminar, I will speak about most about how we are learning about the management of the post-larvae, because of difference from the abalone, and the study of the life, of the post-larval life of the limpets is almost unknown in a worldwide basis. In this presentation, I will start speaking about the limitation of the limpets as the animal itself, how we can directly manipulate the post-larvae, how you can indirectly manipulate the post-larvae, and I would like to finish with a hypothetical method that could be useful to grow out the limpets post-larvae. Okay. How to grow out limpets? In short, nobody knows, because traditionally in the scientific literature, you can find that the settlement requirements and the settlement methodologies were unknown for this animal growth. In general, it was not possible to obtain post-larvae in enough quantities to study the requirements and start the grow-out experience. And fortunately, in the last year, in the AquaInvert project, we were able to start producing post-larvae, and we are starting to understand what this animal will require to start grow-out techniques. The first limitation we have seen in the limpet is that the cell is larger than the soft body. This means that the animal didn't have a body extension like a long neck or appendages to access the food. You need to think in the cell like the animal barrier that will limit the access to the feeding strategy of the animal. Moreover, the height of the animal is also very short, so you need also to consider that the food cannot be provided in a three-dimensional way, because, as we will see in continuation, the motility of the limpets is reduced to a bidimensional environment in either the post-larvae as well in the adults. This means that we cannot think in volumes rather than areas on surface. So we are growing limpets, we are managing surface, not volumes of water. Moreover, the limpets also show a strong attachment. This is evident in the case of the adults. You need special tools like a blunt knife to detach the specimen, but it's also important in the case of the post-larvae. You cannot detach the post-larvae easily. If you directly manipulate it, it's possible, but tricky, and the animal are especially delicate. The cell is very soft. We try to use water pressure, for example, using a pipette. We try to use diluted ethanol, and still the animal remain attached. So instead of moving the animals, you need to move the surface in which the animal is placed. The next thing that we learn is that limpets love the vertical surface. It's commonly observed in the adult tanks, but in the case of the post-larvae, it's also very common to observe this behavior as well. we try to culture the postlarvae in Petri dish, and almost between the 80 and 90%, considering 100 of a specimen, is always located in the walls of a Petri dish. It's very uncommon to find a specimen located in the bottom of a Petri. So we can extrapolate it on a culture system, and we can think in vertical surface to grow out these animals. An associated problem is that the limpets commonly move above the water level. The others had a special resistance and mechanism to survive this condition, but the postlarvae no, leading to the risk of desiccation. It's not working. I'm sorry. Yeah. The major cause, the greatest cause of mortality that we found in our experience managing the postlarvae is the desiccation due to the postlarvae that move above the water level. And it is a thing that we need to consider to start the grow-out experience. And finally, and maybe it's a problem that we will need to solve in the future, and sorry, because this is not working as it should. No. The limpets are unable to recover its normal position if we talk. In this case of the adults, this all never happens unless you place the animal in this position. But as you can see in this video, it works fine, please. Sorry. I don't understand. The computer is not working well. I apologize for this situation. It's not working. I will try to stop sharing the screen. I will say again. Presentación. No. Vaya. Vale. Okay. And these are typical odor movements when it's place of tube, and they will be unable to recover its normal position. In the adult, this never happens because the adults attach very strongly to the surface and the substrate that you use. But in the case of the juveniles, it's a very common accident. And you can find the postlarvae struggling again and again, trying to recover its position, and they will never be able to recover it. How to solve this? We need to think about this problem and how it can be solved. It is an accident, and it's common to find some specimen in this condition, especially if you managing a good quantity of a specimen. Regarding how to direct manipulate the postlarvae, we can use thin needles. And we can use the needles to clean the specimens, to help them to recover its normal position. The problem is very useful at laboratory scale, but for production is not recommended because if you are trying to manage 1,000 specimens, this method is not productive enough. But anyways, it's a tool that can be considered for experimental purpose. Another possibility is to try to pipette the specimen. You need to cut the tip of the pipette, and you can manipulate each individual easily. This method is also useful to manage oocytes, sperm, all the larval stages, and as you can see, the postlarvae. Is another tool that we had to manage the animals, and it's another possibility to take into consideration. But it's better to indirect manipulate the postlarvae. As we explained in previous sessions, I would like to remember the element induced that we found for this limpet species. In Patella aspera, only the crustacean algae are able to induce the sediment thus far, and obtaining a rate of sediment between the 30 and the 50%. In the case of Patella candei, the biofilms naviculum inserta work well. We can induce a sediment rate around between the 30 and the 50%. And in the case of crustacean algae, we can increase this ratio from to around 60 to 90%. Overall, a very good results. And if you had this question in mind, Navicula didn't work in Aspera. We tried it several times, and we still didn't were able to induce a settlement rate higher than 50%, than a 5%, sorry. We select the Patella candela directly on Navicula biofilms in petri dish, for example. For now, this method has a survival for another 1 month and a half. We don't know how much we can extend this experiment, how much we can extend the survival because the experiment is still ongoing. Eventually, we can change the water and new algae. We can recover a few specimens that for some reason dropped from the plate using a maze, and can be recovered the larvae and placed again in the petri dish. And the major cause of mortality thus far has been the desiccation, because, as I explained before, the animals tend to move above the water level. Patella Aspera is tricky, very, very tricky, because they don't like the Navicula biofilms, but you can use small pieces of crustacean algae to induce the settlement, and then we can place the petri dish. We had here a picture of a lot of 6 early post larvae, and we can place them in a petri dish, for example, with a biofilms. Eventually, the post larvae will grow, will develop, and start moving, and will reach the biofilm. And in that moment, the management is identical with Patella candela. The animal will start grazing, will start growing, will move to the vertical walls, will reach the above water level, and we can die desiccated. We need to improve the system, but still we see that we can feed these animals more or less easily, but we need to improve the methodology. How? Maybe this method adapted from abalone might work. I think in parallel sets to increase the production, because we like to better efficiency methodology to use the space. So we need surface in series to use the maximum volume possible. In vertical surface, that must be placed totally underwater, because the animal will move outside the water level and will desiccate. The seeds should be covered with biofilm, and regarding the bottom from the up to the specimen, we still need to find a solution. The first coverage for the seeds will be coralline algae for Patella aspera and Navicula biofilm for Patella candela. Even if possible, in the case of the coralline, to use patches in the seeds of coralline algae, and then these patches can be placed in an area surrounded from Navicula. This will be interesting to test. Also, we can, as the animal grow and survive, you can add additional algae, especially biofilm formers and probably Ulveralens, that works very well in limpets as well in abalone, have a great potential. So as conclusion, you can see that the limpets has several limitations with movement and ability to reach the food. The direct manipulation of the post larvae is possible, but it's tricky and must be realized carefully. To start the culture of the limpets post larvae, we need to start with the incrusting coralline algae in Patella aspera and Navicula biofilms for Patella candela, and the growth system adapted from abalone probably will be enabled to close the life cycle of the limpets for the first time and start the aquaculture production of the species. Thank you very much for your attention.
Thank you very much, Diego. The next part of the presentation, focusing on Patella, will be given by Mirko De Girolamo. He's a researcher from the University of Azores, and his research focuses on abalone, but also on limpets, and he's going to present today some of the research that they've done on aspects related to grow out. Mirko, whenever you're ready, the floor is yours.
Thank you. Thank you, José, for the introduction. have plenty of, okay. Are you seeing the screen? Yeah, I hope so.
You might have to change the view because now we are seeing your slide and
Okay.
your notes.
Okay. Maybe I know where it is, this two. Okay. Is it good now?
No, we're not seeing the presentation.
Ah, okay, sorry. Also, what I am seeing is different from what you see. Let me try again. Okay. Share the screen. I'm here.
Okay.
Okay. I think we are there.
Perfect.
Okay. So, thank you, Alexander, for the introduction. Today, in parallel with Diego, I will talk about the limpet as an opportunity for aquaculture diversification. Actually, I should say another opportunity for aquaculture diversification, because as we have seen during these webinars, in the framework of AquaInvert project, we are also interested in abalone, Haliotistubercula. We have seen also Ricardo presenting the sea urchin. So this is another species for the diversification of the aquaculture. Why is so important diversify the aquaculture? Well, there are many questions, but mainly because there are some factors that can affect aquaculture production, like climate change, ocean acidification, disease outbreaks, and so on. And these factors can actually affect the production of the aquaculture. Moreover, diversified aquaculture means more efficient production through, for instance, polyculture like IMTA, as we have seen in the previous presentation by Maria. And this can reduce both biological and financial risk related to the aquaculture. This is a worldwide strategy to increase strategy, as we can see in this figure. In the last 10 years, we are all around the world are more than 400 species which are now cultivated in more or less intensity. There are other reasons, because for our project is the limpets are interesting, and one is because it's a really good species, is a delicacy in the traditional cuisine in the Azores, but also in Micronesia. Is a sensible species to the fishing. In the past was overexploited, inducing a reduction in local population and an increase in genetic erosion of this population. So we are thinking that we can promote the aquaculture of these species, not only as a source for fishery diversification, but also to relieve fishing pressure through, for instance, restocking plans or restoration plans. So back to the earth, as we have seen in the previous seminar, we have performed a lot of research on different stages of the life cycle of these species, with the aim to close the cycle. We have done, as you have seen by the presentation of Diego, a lot of work on fertilization, on the larval stages and optimization of the settlements, and some experiment on the early growth of these species. Still a lot of work to do on the rearing and grow out systems to close the cycle. Anyway, the idea, the potential protocol, production protocol that we are thinking about include these main stages, which are common to the other species, but they have a specificity typical of the Azorian limpet or the black limpet. So looking at the detail, we are thinking of this protocol will include a stage of for preparation of the plates with biofilms that at the moment, as you have seen by the presentation of Diego, will be probably navicula, but also amphora, and this take almost 2 weeks to get the plate with this kind of species. Other potential biofilm will be the green algae with ulva and ulvella. This process will take almost 4 weeks. And finally, the most interesting and one of the best candidates for the biofilm will be the Crustulus coralline algae. But at the moment, we don't know how long it will take to get this biofilm on the plates. So we are thinking about, because the Crustulus coralline algae are slow growth algae, and we are thinking about weeks or months to get this plate. Anyway, we start, the idea is, of course, as always, to start with the spawning. The gametes will be extracted, or we will get the gametes using different methods. At the moment, we are considering biopsy, dissection, and introduction. And the gametes will be used for the fecundation, and in about 72 hours, we are thinking, at the moment, the data tell us that we are able to get larvae like this, competent larvae, pedivaliger, able to settle down on different substrates. So the next stage will be something like this, as Diego was tell you, with different plates. Of course, the substrates on the plates will be optimized considering the species, but at the moment, with Ulvella, we saw that at least in 5-6 months, we can have animals about 10-16 millimeters. Why focus on this size? Because for similarity with the cultivation of abalone, this is the size in which there is the switch on other kind of feeding or system. So the juveniles will be transferred to other system or to other feeding system or other food. So at the moment, as you have seen in the previous presentation, this is the stage in which we need much more imagination and much more work to do. Because one of the problems we have to solve is the handling the specimens. The juveniles are really, and the adults also are really attached to the wall. So we need a system which give us the opportunity to detach easily the specimens. At the moment, the idea is an indoor system with tanks with a polyethylene liner or suspended containers with a soft mesh, on which, and in both of these systems will be is easy to detach the animal. Other problem we have is feeding at this time. In the case of polyethylene, you see how the biofilm can grow on them. So at least leaving the liner outside in tank just to form the biofilm will be not so complicated. In the case of the suspended containers, we have the opportunity to develop artificial feed. This artificial feed is has to be soft, to be able to be attached to the wall of the container, and be available for the grazing of limpets. And finally, we'll have to, considering the efficiency of the feeding, we will have to consider how many individuals we can culture in this kind of system. So, resuming, we can say, and in more optimistic way, that we can imagine that in almost 1 year we can have individuals of the right size to be used in restocking or restoration plan, and and in 3 years at least get product ready for sale. Of course, with all the consideration, we need to consider this is just an idea we have, and a lot of these stages and step will need to be optimized, improved, but in general, the general vision will be almost this one. With this picture, I leave you, and thank you all for your attention.
Thank you very much, Mirko. Very interesting, and finalizing this limpets part of the webinar. The next presenter now, who will close this webinar, will be Ricardo José. He's from the RDT Center in Madeira also, and his research is focusing on sea urchins. And today he's gonna give us some more information on the nutrition and grow out experiment that they've tested in order to feed their sea urchins. everyone, please, if you have any questions, remember to put them in the chat so we can continue the conversation once the presentations are over. Ricardo,
Okay.
whenever you want, your floor is yours.
Okay, thank you. Let's see if I can start this. This one goes here. Okay. Okay, are you seeing it or? I know it's this one. Okay, is it on or? Okay, sorry, fast. Thank you, Jacinto, for the introduction. As before, my work has been focusing on sea urchins, and today I'm going to talk about grow out and settlement techniques. In the previous webinars, we have already talked about feeding, broodstock, feeding, broodstock induction, and all the other stuff. So right now I'm going to focus on a more technical part on sea urchins. Okay, sea urchins are considered delicacy, and its consumption is around 105,000 tons per year. The reasons for production is mostly due to overfishing and market demand. And aquaculture began in 1960 in Japan with these 4 species that are indicated here. The first hatchery was also in Japan in 1984. For induction, we need usually specific cues for gamete production, like temperature, photoperiod, and diet, which is something that we can manipulate in hatcheries. So it's rather easy to do, as well as it's not difficult to keep sea urchins in tanks. Rearing time is from 2 to 5 years, and mostly until now, production in Japan was for restocking, in which they had several areas where they would remove predators so that the sea urchins could evolve in good conditions, as well as increase the algae presence. In Canada, the sea urchins has been a product of the Aquaculture Association of Canada. They have several works published concerning gonad enhancement, manipulation of reproduction, cost-effective grow-out systems, seed supply, as well as management of the resource in the wild. Okay, this is more or less of a tank system used in several articles for production. We usually have a sump, a skimmer, a chiller to induce the temperature, and several tanks where the sea urchins are stocked. This is not much different from this system. It's only a matter of increasing the individuals and the use of a heater instead of a chiller. This is the system that we currently have at the center. On the left, you can see a set of 4 tanks. The red ones are our broodstock, and the white ones are the ones that are meant to use in rearing sea urchins. On the right, you have a system of the PVC plates that we plan to use for settlement of sea urchins. These plates are a gathering of information from several papers in which the authors usually mention that they use several plates similar to this ones to production. For row enhancement, which is another part of the sea urchins, usually it's some nets in open waters, like the sea nest crates that's used in Norway, Japan, and Iceland. They place several sea urchins and let them grow on these nets, and that's it. I know it's short, but it's mostly because our work is focusing mostly on trying to grow out now, and due to the COVID, we were not able to. Our work plan is a bit delayed. Thank you.
Thank you, Ricardo. Okay, now, I mean, it seems that we have quite a lot of questions. So I will start with some of the questions that were asked to Mappy regarding the abalone. And the first one is the Ipnea production in for the algae culture. And the question is if the production was done using vegetative reproduction, or if it was done using germlings production through spawning.
Well, thank you for the question. No, all the macroalgae that we grow in our facility are asexual, in a asexual method. We just inoculate them every 2 weeks. We harvest and we use part of the algae of the biomass to feed the animals, and the rest, in the case of vulva, the stocking density is 1 or 2 grams per liter. It depends on the moment of the year. And the sanction that it's going to receive, because we want them to sporalize. And in the case of Gracilaria, we have done several studies, so it can be between around 6 or 8 grams per liter to inoculate in the tanks, and then they just grow, and we harvest every 2 weeks. Every 1 week, we just take out a little bit of biomass to feed the animals weekly with both algae, but then the rest still grow, and we have kept them for now in those tanks for 5 years, and with a quite good growth. Despite it depends on the amount of nutrients that we have, because as I said before, we work in an IMTA system. So they do receive the nutrients that comes from the rest of the facility. So if we are working with a lot of different, if there are a lot of trials that are taking part in the facility, so the amount of nutrients will be higher. But if there's not, so they are going to receive less nutrients. So indeed the protein content, it's going to change in that sense, maybe just 15, 17. That I would not say that they are enriched, but they can reach up to maybe 25 or more than that if the nutrient that they receive is higher. Okay, I think I have answered more than I was asked.
Yeah.
So, thank you.
Thank you, Mappy. Related to this, the question is about the feeding interval that you're using to feed the abalone when they are produced in offshore production. How frequently they were fed.
Sorry, they were fed weekly during the trial. Okay.
Okay, another related question to the nutrition is regarding when the animals were fed with the compound feed, how was the water stability of the pellets measured?
Okay, that's a good question because we are always working with the water stability of the diets. You know that abalone, they eat mainly during the night. They've got a nocturnal behavior. So if we feed during the day, we need at least the diet to be stable for 24 hours. You know? The commercial diets, yes, we have done stability testing where we do it just into the tanks. We put the diets just like we would do in a trial, but without animals, you know. So we weight the in it's what we call, you know, the weight of the diet, and then we 24, we have done 12, 24, 48 hours after we remove the diets, and we try them and we compare what happens. With the commercial one, they can be stable for 24 hours later, maybe around 90%. So they just, the leaching is just 10%.
We lost the connection.
Around 17 hours later. So we really have to improve the water stability. With the formula that worked the best one, you know, the one that has Palmaria, we have sent it now to a foreign farm, not farm, enterprise to process it. But it's still, we are testing it now, still it's not well binded. So we think that we cannot feed with the diet longer than 22 hours, 24 hours, because it will not work. I don't know if I have answered.
Yes, thank you, Mappy. You've answered the question. And the last question regarding the abalone and linked also with the diets is if you've observed or you've done some studies on the difference in taste, consistency, color, smell of the abalone after they've been fed different diet.
Yes, we have indeed. In June, yeah, after a 6-month trial, when we fed in a let's say pilot scale, we fed abalone during, I think it was 4 or 5 months with 2 different feeding regimes. The first one was what we called mixed diets, meaning that they were fed 4 days a week with a formulated feed, indeed a South African one, commercial one, but feed, and 3 days, Friday and the weekend, with a fresh algae. That was the mixed regime. And the other one, they were fed just with a formulated feed, the same one, of course. So we wondered if we were going to find any differences in the quality, we mean the flesh quality of the animals. Regarding growth, those that were fed the mixed diet, it was significantly different from the one that from those that were fed just with artificial feeds. That took, I think, 2 months more to get to the commercial size. So that's the first issue. But we performed a tasting panel with 180 people. We gave them both origins, those that were fed with just fresh algae. No, sorry, not fresh algae and artificial diet, and the other one. And they all had a very high appreciation. And it was quite interesting because we made some tricks, you know. Some of them, they knew what were they eating before they ate them, and some of them they knew it, but later, about after they have, and they have to answer a questionnaire. So sometimes when they knew that they were going to feed to eat abalone that have been fed with the compound diet, the presentation was not as good. But finally, they were, I think, they have to point between 0 and 10, and the overall appreciation of the animals was quite near 10. So a very high one. We were very happy with that. And regarding the differences, I think that those that were fed just with artificial feed, they were a little bit softer, and the color, it was whiter. The other one was yellower. But in general, there was not a negative persistation about the animals that ate the commercial diet. Indeed, we know that the only way that abalone is going to be possible to produce here in the Canary Islands, it has to be with a mixed diet. We mean both diets. Because, you know, the food conversion ratio, if you feed the animals with just algae, if it quite enriched, I mean, 30 or 25% of protein, you can reduce the food conversion ratio down to maybe 14. But anyway, you're still needing 14 kilograms of food per kilograms of abalone. That means a huge amount of food. So, it's not an easy way to handle. If you have to produce, if you compare with France or, you know, places where you can harvest the wild macroalgae, yes, maybe you may support this kind of production, but not in the Canary Island that 100% of the algae has to be cultured. So we think that what we will do is, if these get to the market, or if there is an, you know, a potential farm, they should feed with both diets. Fresh algae is not only good for growing, but for healthier animals. It has a lot of property that the compound feed probably don't have. So I think that a mixed feeding regime will be the proper one. Thank you.
Thank you very much, Mapi. And I think we are done with the questions regarding the abalone. And now we'll move to the questions regarding the limpets. So the first one is for Diego. And the question is, if you think that the substrate, the rugosity of the substrate could have an effect on the fixation of the larvae. I mean, not only the biofilm that you are using, but do you think that if you would use like a similar biofilm on two substrate that would have different rugosity, do you think the rugosity would have an effect on it or no?
Maybe, but I'm not sure, because the major problem with biofilm stands from granulates formation, because when algae tend to grow over other algae and is detached. I think that is the major cause of the detachment of the specimen. I think that the solution is going to a better formation of the biofilms and the improvement of the biofilm maintenance. Okay. I think so.
Because the crustoscorolline algae is, the rugosity of the crustoscorolline algae is way higher than the one of the diatoms.
Yeah, the problem with the diatoms is that in the biofilm I observed that many of them start to grow one cell over another, and then this fragment detach. And I think that in some way the limpets are raised to avoid formation, and with the algae detached, the limpet detach with the macro accumulation of the atoms. In the colonies, I still didn't find this behavior because we still didn't use the codalines in vertical surface, so we need to try it to know to see what happens. And another problem is that sometimes the juveniles manage to time to reach the water level and float, and in some moment they drop.
Okay.
So maybe because they try to move and thanks to the water tension, they reach the water surface and this moment loss of contact.
Okay. Thank you, Diego. And then related to this also, there's a question for Mirko, and they're wondering if you have tried using chemical components to allow the detachment of the post larvae when you want to move them around.
Yes, one of our first trial was with how it's called olio di cravigno, is clove oil, okay. And because we were used to use this kind of chemicals with juveniles of abalone, and it works. But anyway, we did this experiment trial with the adults. So probably the effect of the chemical is different. It works, but it takes much more time than we expected. And as far as I know, the reason of this is because the attachment of limpet is not only a physical attachment. Is not just the food that make a suction of the air, but is also release a chemical compound that favor the attachment to the wall. So probably is not enough to anesthetize the animal, but it will be necessary some chemical that affect this biofilm that they release on with the food.
This was tested on post-larvae or only on the adults?
Only with the adults, but I'm pretty sure that you can use it, and probably we will try also with the larvae, because with the juveniles, I'm thinking when I say juveniles, I say individuals of around 10 millimeters. It works well. It works well with the abalone, and yes. So I'm pretty sure that will work also with the limpets.
But in that case, if you would use it on the post larvae, you would obviously have the post larvae falling on their, flat. And then would this be an issue of what Diego was commenting before, that once they are on their shell, they don't have the ability to get back on their foot again? Question. Yeah,
of course. If we think about this method just to detach the individuals and then put them in some surface or another surface, this can work. Of course, you need a lot of manual work, because it's not like a baloney that even if he's upside down, is able to get back on the right position. In this case, you need to pick each animal and put in the right position on the right surface. So yeah, it's not so, is a little bit tricky. Other way we test can be to just leave the limpets to move in other surface, much more attractive, with with the biofilm more interesting for them. So this can be a kind of tricky to get the larvae in the right place without manipulation.
Yeah, maybe one substrate for the element, another one for the ground. When the animals you'll decide to move.
Yeah, exactly.
Thank you, Mirko. There is another question for you. It's if you can indicate how is the grazing rate and the maintenance of the animal on the polyethylene liner in comparison with the mesh structure that were used in the suspended structure?
No.
What was it different, or
No,
was it?
no. This is just what we are imaging. This is a protocol that is on the air because we have animals on the plastic liner. We can measure in some way the rate of grazing, how much surface is grazed during a week, but I'm talking about adult specimens. So what we need to know now is okay, once we put the animal in a system like this, which is the feeding rate of this animal, and how many animals we can manage on this kind of.
But you find grazing marks on the.
Yeah, definitely. Both on the liner and on the plate.
But
What happened with the macroalgae? You observe they are also grazed or only the films?
You talk about macroalgae.
See the macro, the big ones. You observe some grazing or only in the films of the lines?
No. In the macroalgae, when I talk about the macroalgae, is the journeyings of the vulva, and you can observe some marks. But of course, this is a system, is a mixed system, because we don't have just macroalgae on this plate.
Yeah.
We also add the atoms, so probably is a mix of
Yeah, my question is, in what system in what kind of algae you observe more grazing behavior? In the biofilms or the biofilm-like or macroalgae-like?
No. I think that the ideal is the biofilm. The biofilm has the structure is much more available for the grazing behavior of the limpets. But of course, I think that we need also to think about some macroalgae, because they eat them.
Yeah, especially the bubbles.
Are you able? We have some problems of connection.
And but for the moment, the grazing rate that you've measured is only for adults, and in which substrate? If it's not on the polyethylene liner, on which one was it measured?
Okay, no, the rate, we don't have, actually, we don't have a number. I see we have the plate, and we can observe the grazing on the plate, as Diego is saying, the marks on the grazing of the plate and on the liner. So now the idea is, okay, put that to get a zero image of the linear, and then see how long does it take for individuals of 6-8 centimeters of shell length to eat a kind of surface. Yeah, this is the plan, but it's something that we would like to realize, to know.
Okay. And we have a question that's for Mirko or Diego, both of you. The question is about the commercial size of the limpets. At which size are they commercialized? And is the size the same in Azores and Madeira, or is it different?
Here in Madeira is between 30 and 50 millimeters, tend to be less than 50 due to the exploitation. The limpets have not the opportunity to grow much of this size. So we're speaking around 40 millimeters, and it's around 2, maybe 3 years, yeah, as Mirko said before.
Yeah, in the Azores, something like that.
Okay, and then, so in the future, I mean, if you would now, considering that you think that you need to look at grow out possibilities, what are you planning to do or to test for the grow out on both, I mean, both in Azores and in Madeira?
Mirko, you would like to start?
Okay. Yeah, our analysis is about individuals that naturally grow on our tanks, and we saw that at least for the black limpet, Patella candei, we get commercial size more than 40 millimeters in 2 years, and this is in just normal natural biofilms. So for the black limpet would be, for the aspera, the Azorean limpets, we don't know, but is what we saw in the at least for the juveniles, the growing rate on Ulvella, for sure. It seems that Patella candei is faster growing than aspera. So this is the reason, because I put there 3 years than 2. But this is just imagination and what I would like to get.
Yeah, I had a similar opinion. Candei is easier than aspera regarding at least the settlement. I think that we like to test the seeds or biofilms for us for candei, and then we can place the biofilm to change the position to either enrich with artificially cultured algae, or either exposed to natural water, to natural enrichment from the algae that come from the outside of the installation. Either way, I think that is the best option for candei. For aspera, I think we will need to place at least the faster approach is using a limpet cell already covered by corallines as a settlement island to obtain the postlarvae. The major problem with this is that we had contamination because we had a lot of microorganisms living in the limpet cell, so we will contaminate the culture, but we would like to obtain. Right now, the postlarvae is the only option that we have. Alternative, we need to go out a place with coralline algae. And in any case, if the coralline is obtained by the natural ones or the cultured ones, I think that we need to place the corallines in the sea of the atom biofilms, for example. So we had a point for settlement and another one for the limpets can move out and feeding. The problem is we to decide we place freeze the element and then we place them with the biofilms, or dealing the corallines with the biofilms. because we didn't need to check if we had antagonistic behavior. So maybe the olefins can inhibit the codaline effect or not. But for now, I think that the short-time solution that we have for this species, and we will find another one. We are taking chemical induction of those elements, but it's very difficult. Didn't work in Candei and in Aspera, we need to see.
Okay, thank you very much. And then finally, there is a question for Ricardo on the sea urchin. And the question is regarding the future experiments, the grow out experiments that you're going to perform. What are you planning to perform for to test for grow out of your sea urchins?
And to test in relation to what, systems or feeding?
Both.
Okay, we're going to try one system, the one that I showed you, because it's based on the literature and it's the most suitable. And we are planning on adding microalgae as well as natural biofilm that will grow. The biofilm eventually will be identified as we test and see what grows and how the sea urchins grow.
Are you planning to test any artificial compound feed?
At this moment, no. We eventually will try the feed that we are giving the broodstock that it's maize, to see if at which point they start accepting it, because it's a good feed.
And there is another question that just came thank you very much, Ricardo. And there is another question that just came in for regarding the limpets. And it's, do limpets have like a stacking behavior? You know, I mean, do they tend to go one on top of the other? Or do you observe that they tend to stay always on the same place, that they have a homing behavior? So stacking and homing, have you observed this?
Depends, because according to the ecological studies, the limpets are very territorial among them and also had a homing behavior, so they will maintain the distance. And the juveniles, for now, looks like they can tolerate themselves and can stay together, but they will not stay. But maybe it's possible to obtain post-larvae growing over turtles. It's a possibility, and in some species happens. I would like to release a short test in this week, and we will see what happens. But theoretically, it will be possible to use adults to induce over tercel. But we will need to check it.
Sí.
Yes, I was wondering if they have that behavior, because we do observe it in abalone. And that's why the socket density is so important, because it's not a problem of having enough food, but to be able to get to the food, because since they are stacked and when the socket density is high, so it's not so easy, the one that is down or in the middle of the mountain, you know, yeah, to be able to go out and to access to the feed. It doesn't matter if the feed, you know, if they have feed enough, but they cannot get there. That's why it's an important issue.
Well, it's interesting. We sometimes, it's not very common, but you found some juvenile with a maybe 5 millimeters length or so above a set of a larger specimen. And you can see over the homing scar of the specimen. Yeah. So happens, the will happens.
Okay.
But we need an important difference inside for now.
Okay.
Okay, well, thank you, everyone. We've gone over the time that we had allocated. It was a very interesting exchanges. I would like to thank everybody for their participation during the series of webinar that we've done, that went from broodstock to larval production to post-larvae, to grow out, to be able to go through the whole life cycle of these wonderful invertebrates that we like to study. It was very interesting. I don't know if we're going to be able to do other online webinar. We'll still have to see what will be done. But anyway, in the meanwhile, I would like to thank all the participants and then all the attendants that were patient enough to listen to all these information. The information is online on the website, so people can have access to the different webinar that took place. So keep on having a look at it. And if you want to keep on being informed about what we're doing on invertebrates, and you can also have some information on the webpage. Thank you very much and hope to see you soon.
See you.
Goodbye.
Thank you.
Bye.
Bye.
Bye.